Welcome to Copernicus AI, Frontiers of Science. I'm Matilda, and today we're delving into a truly foundational breakthrough that revolutionized our understanding of gene regulation: the ability to identify protein-binding sites from unaligned DNA fragments. Adam, what makes this research so profoundly revolutionary, a true delta in scientific thinking?
Thanks, Matilda. We're going back to 1989 to the seminal work by G.D. Stormo and G.W. Hartzell, published in the Proceedings of the National Academy of Sciences. Before their paper, Identifying Protein-Binding Sites from Unaligned DNA Fragments, the link is in the description, researchers faced a major hurdle. To find the specific sequences where proteins bind to DNA, which are crucial for controlling gene expression, you typically needed to align multiple DNA sequences that were already believed to contain these sites. This pre-alignment step was a significant bottleneck, often requiring prior knowledge or strong hypotheses.
So, if I understand correctly, the paradigm shift here is moving from a process that needed existing, aligned reference points to one that could essentially discover these critical patterns from scratch. Without that prior alignment? That sounds like a massive leap in what was computationally possible at the time.
Precisely, Matilda. It was about recognizing subtle, recurring motifs within what was often a sea of noisy genetic data without a perfect template. The conventional understanding limited discovery to areas where we already had some genomic context. Stormo and Hartzell's method elegantly bypassed that by using statistical inference. They developed an algorithm that could identify these short-conserved recognition patterns within a set of diverse unaligned sequences known to bind a particular protein. This ability to extract meaning from disorder was a game-changer, opening up vast new avenues for genetic analysis and discovery.
That's fascinating. It really challenges the idea that you need perfect order to find patterns. How did this initial breakthrough evolve, and what were its immediate implications for the scientific community?
The implications were profound. It meant we could start to systematically discover novel binding sites rather than just confirming known ones. This was absolutely essential as large-scale DNA sequencing projects began to ramp up. Following this, the work evolved. Just a few years later, in 1992, L.R. Carden and G.D. Stormo, again collaborating, extended this with an expectation maximization algorithm for identifying protein-binding sites with variable lengths from unaligned DNA fragments, published in the Journal of Molecular Biology. This advancement allowed for more complex binding sites with variable length spacers, further increasing the method's utility and predictive power. We'll link to that in the description.
So the core idea was refined and made even more versatile. Beyond identifying the sites themselves, how did this fundamental understanding contribute to a broader picture of how DNA-binding proteins actually function within the cell?
It laid crucial groundwork. Understanding where proteins bind is intrinsically linked to understanding how they bind and why it's so efficient. For example, later research like Rajamanikam Murugan's 2014 paper on the mechanism of rapid binding of transcription factor proteins at specific sites on DNA, available on arXiv, explores how these proteins locate their targets much faster than simple 3D diffusion would predict. Our ability to pinpoint binding sites informs these deeper mechanistic studies, allowing us to ask more sophisticated questions about cellular machinery. The original work helped establish the molecular addresses for these critical interactions.
That connects the what to the how and why beautifully. And with the explosion of genomics and bioinformatics, how has this foundational concept of identifying binding sites without alignment paved the way for modern computational biology, especially with machine learning?
It's been absolutely foundational. These early statistical methods were precursors to today's sophisticated AI and machine learning approaches. The challenge of finding subtle patterns in vast biological data remains, but the tools have evolved dramatically. For instance, Sokina Kataune and colleagues published Efficient Prediction of DNA-Binding Proteins using machine learning in 2012, highlighting the shift towards algorithmic prediction. More recently, in 2023, Yufan Liu and Boxwei Tian explored protein DNA binding sites prediction based on pre-trained protein language model and contrastive learning, leveraging cutting-edge AI. These developments, which we'll link to in the description, build directly on the principles established by Stormo and Hartzell, the idea that sequence information alone holds the keys to understanding these interactions.
So we've moved from clever statistical algorithms to leveraging vast datasets in AI to make predictions with unprecedented accuracy. What does this mean for practical applications, for investors, for medical professionals, or just the curious mind wondering about its real-world impact?
The practical applications are enormous. Accurate identification of protein DNA binding sites is vital for understanding disease mechanisms, as errors in these interactions can lead to conditions like cancer. In drug discovery, knowing these sites allows for the design of molecules that can modulate gene expression, either activating or suppressing specific genes. It underpins synthetic biology, where we engineer new biological systems, and is critical for advancing gene editing technologies like CRISPR, where precise targeting is paramount. It's fundamental to our ability to manipulate and understand life at its most basic level.
Adam, this has been an incredibly insightful discussion. From an ingenious statistical method to the backbone of modern genomics and AI-driven biological discovery, the work on identifying protein binding sites from unaligned DNA fragments has truly reshaped our understanding of life's blueprints. Thank you for breaking down this groundbreaking research and connecting it to its revolutionary implications.
My pleasure, Matilda. It's a testament to how foundational theoretical work can continue to influence scientific progress for decades, even centuries.